Okay, well, let me thank everyone again, and I think we can get started, and others will hopefully be joining as we get started. So first, I would like to thank everyone for joining us for the July session of the Excellence in Breeding GOBii webinar series today. We're excited to have a speaker who will tell us about a tool that was developed as part of the GOBii project through a collaboration with Diversity Arrays Technology, also known as DART. Andrew, our speaker, is kindly joining us from late on a Monday evening in Australia to tell us about the DarkView tool. And I just wanted to let everyone know that we will be recording this seminar and we'll make it available later for anyone who may want to access it. During the presentation, if you have any questions, please feel free to share them through the chat window, or you'll have an opportunity to ask them directly to Andrew after he presents. So before we proceed, I'd just like to take a moment to introduce our speaker, Andrew Kowalczyk. Andrew studied software engineering and statistics at the University of Melbourne and got his degree there in 2013. He engaged in bioinformatics work in the realm of human genetics in the Melbourne School of Population and Global Health before joining the Diversity Arrays Technology team in 2015. There, he has been focusing on developing applications for downstream data analysis and visualization, including the DarkView tool that he will show us today. I'd like to thank Andrew for participating in our seminar series and for telling us about marker data curation via metadata filtering today. So Andrew, the floor is yours.
Okay, thank you very much, Kate, for that introduction. So as Kate mentioned, I'll do a demonstration today. We'll just start with a few slides just to get the ball rolling. Alrighty. So DarkView is a desktop application. It's a Java desktop application which you download and run on your desktop. It's a plugin which is a member of the KD Explore framework. Some of you may be familiar with it. And it essentially is a tool which works with both Dart reports, and that's in the SNP, Silico, and counts format, as well as HapMap formats, in particular the two-letter codominant, the dominant, and SSR formats. So what is it for? So typically during marker curation and assessment of these data sets, our users would like to get a sense of the distribution of the various statistics across their data, across in particular the markers and samples. And so one of the core features of this application is to essentially automatically generate those plots and give the user a sense of what the data looks like. And so these statistics will include those of the zygote counts and frequencies, things like core rate and minor allele frequency. Another feature of the app is the ability to create, edit, and reuse filtering rules. So being able to subset the data set, for example, core rate less than 50%. And we've also enhanced the ability of creating these filtering rules by providing various visual displays to help users understand what the effects of those rules are. And of course, you'll be able to export a subset of the data set according to those filtering rules. So I'll just jump straight into the demo. It's the best way to show what it's all about. The software is available right now on this URL. I think we'll send out an email with the link afterwards for your convenience. So on the website, we have a link to download the application, which you just click and automatically download. We also have some documentation available to help users understand the various features of the app. So quite quickly, it's downloaded for me. Just open up. It is a zip, so you'll need to extract it. And then going into the folder, And in this folder, you'll see KD Explore starter shortcuts. So to get it going, just open it up and run with your Java runtime. And this will open up the KD Explore framework. So in this bundle, we just have the one plugin for DarkView. Some other variations of the KD Explore application may have additional plugins. This one just has the one. So to launch a plugin, you just click on the plugin you wish. So up has popped open DarkView. At the moment, it's a bit empty. So we need to load one of our data sets here. So to do that, go to Windows, File and Open. And then I'll just grab one of my HapMap data sets and drop it into there. So now that the data set has been loaded, a few things have come alive. On the top left here, we have a statistics panel. Below it, we have a filtering rules. And on the right, we have a graph display, which is currently empty. The top left, we have a display here showing the number of markers and samples. So for this data set, we have about 4,000 markers and 300 or so samples. We have a set of statistics, and these are organized into markers and samples. And for each of them, they're further organized into numeric and non-numeric forms. So we have statistics like core rate, data count, head count in the numeric. In non-numeric, we have statistics like the various classes of heterozygotes, the HOMs, minor allele, etc. Each of these table rows has a set of basic statistics such as minimum, mean, and max. So for example, for core rate, we have a minimum of 0 all the way up to 0.99, and with an average mean of 0.36. We can further expand this out to get a greater number of stats, and this will basically present all the box plot-like statistics like interquartile ranges, variance, and standard deviation. You may have noticed that I had prematurely clicked open the graph. This will essentially open up a graph which will give us a scatter plot display of the distribution of this statistic. So what has happened is this is a statistic which has been calculated by the DarkView. And so for every single marker, we've calculated the core rate, and then we've sorted that in a descending order and displayed that in this graph here. Just move that out of the way. So you can see that there's a sort of multimodal distribution across the marker core rate, and so I'll just be exploring that in today's session. Just to finish off, for the non-numeric types, you can also open up a display. Obviously, for non-numeric, it doesn't make sense to plot it in a scatter plot. Instead, we just have a table presenting the unique values. And likewise, for sample statistics, we can also open up a plot to get a sense of the distribution of the statistics. So for example, for sample core rate, we can see a relatively low core rate, and again, some sort of multimodal distribution going on. So typically, because this is a data set produced by Dart, typically we would expect to see something of an exponential distribution, not this multimodal. So we'll have an exploration, we'll explore what's going on here. Another feature of the DarkView is the marker and metadata view, opened by clicking this button here. And essentially, what this window presents to us is our entire genotypic data color-coded by the various alleles. I can choose to reorganize the coloring rather than having this sort of rainbow coloring effect for every single two-letter representation, I could choose to color it by the ref or alt as described in the HapMap file, or I can choose to color it by minor allele. So for example, for the minor allele, for each marker, I'll color with green. The alternative will be a blue, het will be yellow, and missing will be white. So if I click that, I'll get a more consistent appearance of the data. I can choose to zoom in and out of this data set by holding down Control, and it'll essentially zoom in where my mouse cursor is. There is also an ability to zoom in and out via a menu, and I can zoom to max, for example, and I can have a look at the data at maximum. At any time, I can just reset the zoom by clicking this magnifying glass. A component of this marker and metadata view is the ability to plot the various statistics alongside the data. So for example, if I'd like to show the marker statistic for core rate, I'll just click this button here, select the core rate, and then click OK. Essentially, this will just plot the values of the core rate for every single marker alongside my genotypic data. Likewise, for samples, I can do the same. And so I'll choose core rate again. And so I'll get a display of the sample core rate alongside. To make more sense of this data set, I can then choose to sort it. So for example, I can click this button down here. That will sort it into currently ascending order. And I can cycle through various sort orders again. So now I've resorted it into descending order. And so we can see that towards, as we would expect towards the top, we have a more filled in color to represent the high core rate markers, down to the bottom where we have low core rate, where we see mostly white, which represents the missing values. Similarly, for the samples, I can do the same. And so I can sort by ascending or descending if I so wish. So you may have noticed that now that I've sorted this data a little bit, we see some sort of segregation occurring here. If I was to guess, there is some sort of population, two distinct populations in this data set. And so I'd like to explore further what this effect is. Because I didn't have any population data information in my HapMap file, we have a feature which will allow you to import additional metadata. So for example, I have a CSV here, which contains, for every simple sample identifier, it contains a species mapping. And basically, I would like to now import this into the DarkView and just see whether this explains this the segregating population. So to do that, back in the main window, just move that out the way again. Click open Windows, import, and additional sample metadata. And then I'll just scroll, find my file, and then click open on that one. So here we have just a basic import wizard to help understand how to interpret this file. You may have seen this in spreadsheet programs like Excel. But essentially, this is a comma-separated file. The encoding is just a standard encoding. There is no header in my sample meta file. And I need to select which column specifies my identifier. In this case, it's column A, so I've just clicked that there. And I'll be importing against the sample identifier, which is the existing field in my currently loaded into DarkView. So just click next on that. And this is just a little simple dialog asking which columns would I like to import. So I'd like to import the column B. And I'll just rename that to something a little bit more informative. And click import there. So my new metadata has been loaded in. If we go to the samples and non-numeric tab, we can see that we've now got a green row, which represents an imported field. And if I just open that up, I'll get presented with the unique values for that metadata. Back in the marker and metadata view, I can now add that imported statistic as a adjacent metadata display. And if I so wish, I can now sort by this metadata, pushing it up to the top to mark that I'm prioritizing to sort by this statistic, and then actually choosing a sort order. And so as you can see here, once I've sorted by the species, we've now distinctly separated the two populations. And so we've got our explanation as to what is causing that distinction. So that's a little brief of the visualization component. But the core function or feature of this application is the ability to actually filter the data. So, for example, looking at this data set, I may conclude that it doesn't make sense to do my downstream analysis on this combined data set. So I'd like to now just separate these two population groups, or these two species, into sub data sets. And so what I can do is essentially, if I'd like to remove one of these groups, I can just check here in the categorical table for species. And basically this will create a filter which will now remove that species from the data set. You may have noticed that the marker core rate graph now has a secondary line. So the original line has now been slightly grayed out. And so we now have a new distribution. And so what's happened here is that the filter has removed those samples which belong to the Chaplin species, and has recalculated each of the marker core rates. And so now we start to see our sort of exponential graph followed by a trailing tail, which now marks the those markers which have a 0 core rate. I can switch, and so I can create whatever filters I'd like. So for example, I'd like to actually filter out Begensis instead, have a look at that. And so here we see an even more distinct exponential-like distribution. Back in the marker core rate, we can apply those filters and have a look at how the data set changes. So if I just max back out, I can just apply the current filters. And so now we've just removed those samples, and you can see alongside the core rate statistic has been recalculated and is on display. I have quite a number of all missing markers or monomorphic markers, so I may choose to remove those as well. So I can go back to the markers tab, and I have a little post filter check here, which indicates that after the filters have been applied, like the core rate or the species, I can then choose to remove introduced monomorphic markers. So if I just check that one there, you can see now that quite a number of markers have now been removed, and now we don't see any trailing 0 core rate markers, as well as there's definitely a number of monomorphs which have been removed as well. You can see the various statistics, how many have been removed. So for this monomorphic filter, we only have, after 3,900 markers, we only have 873 left. 3,000 have been removed. And similarly for the samples, we can see that we have remaining 112, and 212 have been excluded. I just want to highlight that all of these filters are orientated from the context of what we are excluding, not what we're including. Typically, a lot of workstations work in the perspective of what we might want to include, but the filters in this context are all to do with what we choose to exclude. So at any point, I can choose to export this data set and create a new subset. And so I just click this button here. And so just ask me, what would I like to call this data set? So I'll call it Chaplin data set and click save there. Just override the existing one. And so now it's exported, it's created a new HapMap file, and I can, either choose to continue or open it in Excel, or I could reopen it into DarkView. So I'll just reopen it back into DarkView. And so now we see our subset data set matching what we had filtered out before. We see an improvement in the sample core rate. I forgot to mention that before. And so now I may wish to continue operating on this data set and further performing further filtering. And so, for example, I may wish to now create some filtering rules around the various numerical statistics. So, for example, if I want to create a filter for the core rate, I may wish to choose to remove all markers with a core rate of less than 30%. And so here, by clicking on the graph, I get presented with this menu. We have 5 sets of exclude if. These are just the various Boolean comparator operations. So we've got less than, equal, greater, equal, or greater. I'll just go with the less than for now. And so this will create a filtering rule which will cut off all the markers which have a core rate of less than 30%. We can see down in our filter table, we've now created a new filtering rule, which has 767 markers left and 106 which have been removed. You can get quite complex with these filtering rules. So for example, I may wish to create combinations of filtering rules to do with heterozygous frequency in combination with core rate. So for example, if I'd like to now create a filtering rule to remove high heterozygous markers, for example, those which are greater than 0.4, I can choose, click on the graph again, create a rule there. But I may wish to also impose the rule that I only want to remove markers which have a high frequency, but only if they have a core rate less than 0.5. So for example, if I now introduce a core rate of less than 0.5 alongside the het, I'll get asked a little dialogue here just to help explain the filtering principles. Essentially, you have a choice between a disjunction rule or a conjunction rule. Conjunction rule will essentially mean that both or all filtering rules in this category have to be satisfied for the marker to be removed. So for example, we have our existing core rate of, we have our existing het frequency of greater than 0.4 alongside the core rate of less than 0.5. So with a conjunction, we will only have one marker which will satisfy that criteria in this set of examples. So if I click and, we can see that not all of the markers have been removed below this core rate because they may not satisfy the heterozygous rule. And so you can create as many of these filtering rules as you'd like. And so one of the features we have for this is that at any point you can save your filtering rules. So here I'll just save it to file, filtering rules of core rate 0.31, het frequency 0.39 and core rate 0.49. Just save that. So it's just popped open. It's now just saved here. And so this will allow users to be able to reuse those filtering rules in future. So for example, if I was to come back to a new data set of the same population group, in a year's time, I may wish to reuse the same filtering rules which I had used previously, my previous analysis. So I can just grab that filtering rule and drop it into here. And so that will repopulate those filtering rules for my new data set. And so, yeah, at any point, I can just save, again, create as many data sets as I'd like, as many subsets rather as I'd like. And these can be re-uploaded back into the GOBii system. And I believe we'll also have a feature to be able to store these filtering rules as well alongside, so that you can easily retrieve them in future. Okay, so I think that more or less wraps up the core components. I guess we can move into questions.
Thank you very much, Andrew, for a great presentation. Does anyone on the webinar have any questions that they'd like to ask to Andrew? Hope people are able to unmute yourself, but if you're not by any chance, please raise your hand. I see some people have unmuted. Any questions?
Hi, Andrew.
Hello, Andrew. Go ahead.
Hi, Andrew. This is Liz. I don't know whether I missed it, but did you talk about the work we're doing on a GOBii import?
No, I didn't talk about it. Yeah, so that's a good, thanks for reminding me. We do have a feature which is upcoming to enable users to load a data set from GOBii. I think we've more or less got that working. It will be available in the next feature. Unfortunately, this version here doesn't have it available, so I can't quite show it. Okay.
So, I mean, you'll be able to use GOBii calls to be able to pull data from any GOBii-enabled database, like GOBii or Gemini.
Yes, that's correct.
Hello, Andrew. This is Tom. Thank you for your demonstration. This is a very promising tool. Will there be sort of analogous to Liz's question? Besides retrieving data from, do you have in your development roadmap plans to put data back? That if a scientist uses this tool to do the work you've demonstrated here, can the outcomes of that be saved back to a breeding system for use by other functionalities of the system?
So yeah, it's a good question. At the moment, we only have a feature to export to a plain text file. So the assumption was that this file would be taken and then uploaded back into GOBii. We can definitely have a look at extending that to automatically upload to a breeding system. I imagine it would be some sort of API which we would just interact with. Is that what
I would imagine. Yes, some type of API. Just like Liz was mentioning, API to bring data into this, and APIs to, in effect, put data back.
Yeah, we can definitely have a look at that.
This is Palival. My question is how much big file this can be handled? How much big data it can be handled? It has any restriction, or this only depend on the memory or something?
Yeah, it's a good question. Look, to be honest, this version right here doesn't perform very well for the larger data sets. And a large part of the last several weeks, I've been working to optimize on that front. We want to aim for data sets which are, up to 1 gigabyte in size. So that's our roadmap at the moment.
And in future, I think it will be just like the earlier I say, it will be happy because now it is taking the text and CSV format, but after saving, if it can save in the BCF or P-Link or some more demanded format, it will be really very helpful. Otherwise, in the data marker, always this is the problem. They give in the CSV and text format, and changing is always very tough. I must go in the R and do different package, and it killing the time, So really, it will be helpful if we make it from CSV, and then we can save it either in BCF or P-Link format, and that will save a lot of time for all of us. Yeah, thank you.
Yeah. Okay, we'll take that on board.
Yeah, Andrew, we've had that feedback from a few people that even if you import in the 2-row format from Dart, if you could export as a hat map, to have that flexibility to export in different formats would be really good.
Yeah. Yeah, it definitely would provide more versatility in the downstream analysis.
Hi, Andrew. Milka here.
Hi, Milka.
Demonstrating this really cool tool. My question is about the import file. You imported a HapMap file. Well, are there any restrictions or other requirements for which files you can import, like the 1-row, 2-row file formats?
Yeah, so at the moment, it supports just the standard HapMap and the various Dart data sets. I guess Dart because we're biased. So yeah, it's just to reiterate from the slides, it's the HapMap 2-row, the HapMap 2-letter, codominant, dominant, and SSR. And then for the Dart data sets, it will be the SNP, both 1-row and 2-row, the counts format, and the Silico Dart data sets. Did you have any other data sets in mind or formats in mind?
No, just those ones. But
Okay.
you have it in the documentation, maybe with some screenshots of what the file formats should look like.
Okay. Yeah.
Thank you.
Are there any other questions from anyone? And Andrew, I think if it's okay with you, I'm going to go ahead and just paste the link that you showed in the chat box for everyone to get access to the site.
Andrew, just a quick question.
No
How to save this figure? Scatter. We can save it as it is in the PDF or something, a particular format. The figures you are showing here,
Yeah, so you can right click and save it. There's a little function here of feature rather, which will allow you to save it. And so you can then use these in, whatever document. So similarly, we have one for the marker and metadata view. And so typically I'll use this when my boss, Andre, is asking me to inspect the core rate across various data sets. I'll typically save one of these and send it to him to say, hey, this is what it looks like. So we have those.
It will be nice if it has some x-axis and y-axis, what is the, legend, then really it will be helpful for purpose.
Sorry.
I'm saying from the, like I was saying on this x-axis and y-axis, there are some values. So it can tell us what is meaning of this value, like here in the filtering in the marker call rate. Nothing is coming x and y-axis, what are they, So it will help us if it will tell us what is the x row, what is the y row you are using in this. So it will really, we can present somewhere,
Right. So we have some values for the y, but they are missing for x. I assume that's what you're referring to.
And then we should know what is these values, y-axis and x-axis, what they are. It will really help us,
Yeah, that's a good point. I should explain that the x-axis is essentially from the very left to the very right. It's the full set of data points. So for example, for this data set, I have 873 markers. So there'll be 873 points stretching
Yes, yes, yes.
from the left to the right. But yeah, we can definitely pop in an axis to keep track of that.
Thank you.
Yeah, that's a good one. I just realized now I did skirt over that we also have a histogram view. So if you'd like to inspect the data in this fashion, we have a mode which will allow you to inspect it. In my haste, I sort of skipped over that.
And also filter on it, correct?
Yes, that's correct. So you can modify those filters. If I just jump to this one, be a bit more apparent. So this will essentially show what portion of the data set is filtered out according to the binned values. Just close some of the other graphs, make that bigger.
That's good.
Is any other questions?
Hi, Andrew. This is Dark. Yeah, can you hear me?
Hello. Yep.
Yeah. So I understand this is a filtering very nice feature. It's within a data set, right? If you have large populations come from Dart, mixing many different populations, is there a way also looking at them by individual populations?
So in terms of if you had additional metadata.
Right. Yeah. This is by data set, right?
Yeah. So just to understand, so you're referring to maybe population groups which you might have tracked in GOBii and accessible by BRAPI, or?
It's just more generic. If you have quite a diverse populations, your filtering criteria could be quite different. This within data sets may mix, like say, a dozen populations. If you could look at a population specific also.
Yeah, so going back to my additional metadata, I mean, this additional metadata could contain as many columns as you want, and could contain whatever complex groups you may have. And you can, in your filtering rules, you can combine the numerical filters with the categorical. So for example, I may wish to filter by a certain call rate threshold for the Bergensis group, and a different threshold for the chaplain. Is that sort of what you're referring to?
Yeah, exactly. That's very nice.
Yeah.
Yes.
Yeah. Yeah, that functionality is in there.
Are there any other questions? Okay. If not, then I would like to thank Andrew very much again for all of his work in developing this tool, and to everyone who has participated in giving requirements and feedback and testing this tool. But thank you very much for providing this and for presenting it to us today. We will be working to make the recording of this presentation available to everyone. We'll post it on the webinar website. And I also put a link to the software, the chat box, but we'll also make that available at the webinar website for anyone else who would like to access this. So again, thank you very much to Andrew. Thank you to all of you for
Thank you.
And we hope that you can join us for the next seminar in the series in August. Okay, have a good day, good evening, everyone, and best wishes for the well-being of all.
Stay safe, everyone.
Thank you, Kate.
Thanks, Andrew.
Thank you.